on another exciting biochemical adventure with all of you. Riboflavin is that vital component ensuring the uninterrupted flow of electrons, the very spark of life. It's a water-soluble vitamin, meaning our bodies, much like a thirsty patient on a hot day, need a regular intake. We can't store much of it, so consistent dietary supply is key. But riboflavin itself is merely the blueprint. It transforms into two incredible, powerful coenzymes, FMN and FAD, flavin mononucleotide and flavin-adenine dinucleotide. These are the true workhorses, the superconductors that meticulously manage electron transfer in an astonishing array of metabolic pathways, from breaking down the carbohydrates in your favorite biryani to metabolizing the fats from that delicious samosa. FMN and FAD are omnipresent. They are the critical link between the oxidation of your food and the generation of ATP, which is the universal energy currency of the cell. Understanding riboflavin isn't just about memorizing facts, it's about grasping the fundamental principles of bioenergetics that underpin every aspect of human physiology and pathology, something you'll encounter daily, whether you are in a hospital or a quiet clinic in India. Once you consume riboflavin, perhaps from milk, eggs, leafy greens, or fortified cereals, it doesn't just passively drift into your bloodstream. It's actively absorbed, primarily in the proximal small intestine. This absorption process is highly specific and saturable, meaning there's a limit to how much can be absorbed at once, and it relies on a sodium-dependent active transport system. This ensures efficient uptake even when dietary concentrations are low, which is vital in populations with limited access to diverse nutrition. Imagine a security checkpoint at a high-profile event. Only the authorized people, this is the riboflavin, gets through, and that too through a dedicated channel, which is the sodium-dependent transporter. Once inside the enterocyte and subsequently in other cells, the first crucial activation step occurs. Riboflavin is immediately phosphorylated to FMN or flavin mononucleotide. This reaction is beautifully orchestrated by the enzyme riboflavin kinase. This enzyme, like a skilled tailor, takes a phosphate group from ATP and deftly attaches it to the ribotyl side chain of riboflavin. So riboflavin plus ATP gives FMN plus ADP. The terminal phosphate of ATP is transferred to the hydroxyl group on the C5 carbon of the ribotyl moiety of riboflavin. This phosphorylation serves a critical purpose. It effectively traps the flavin within the cell. Unphosphorylated riboflavin can freely diffuse out, but FMN, being charged, cannot easily cross cell membranes, ensuring its retention and availability for further metabolic roles. It's like putting a VIP pass on a Bollywood star. Once it's on, they're staying in the exclusive zone. FMN, now trapped and partially activated, undergoes a second, equally important activation step to become flavin-adenine dinucleotide or FAD. This reaction is catalyzed by FMN-adenylyl transferase, also known as FAD synthetase. Again, ATP is the essential energy donor, highlighting the cellular investment in creating these crucial coenzymes. FMN plus ATP gives FAD plus pyrophosphate. In this step, the AMP moiety from ATP is transferred to FMN, forming FAD. The pyrophosphate released is not just a waste product, it's rapidly hydrolyzed by an ubiquitous enzyme, which is inorganic pyrophosphatase. It is basically converted into two organic phosphates. This rapid hydrolysis creates a significant amount of free energy, making the overall reaction essentially irreversible and highly favorable thermodynamically, ensuring that FAD synthesis proceeds efficiently. This is the grand finale, the complete transformation, making our star ready for the big stage. Now that we have the fully activated FMN and FAD, let's unlock their incredible power. The secret to their electron handling lies in their central isoalloxazine ring system. This unique conjugated heterocyclic ring system is a master of disguise, capable of existing in 3 distinct redox states, allowing it to accept either 1 or 2 electrons, always accompanied by corresponding protons. Think of it as a master jugalbandi artist, perfectly balancing electrons and protons. Now, oxidized form, FAD or FMN, is the electron deficient state, represented by a bright yellow color in its pure form. It's eagerly awaiting electrons like a hungry cricketer waiting for a delivery. The semiquinone form is basically FADH and FMNH. This is where FAD and FMN truly shine and differentiate themselves from NAD+ or NADH. The isoalloxazine ring can accept a single electron and a proton, forming a relatively stable free radical. This halfway reduced state, often blue or red in color, is crucial. It allows flavoproteins to participate in reactions involving free radical intermediates and critically to bridge the gap between 2 electron donors like NADPH and 1 electron acceptor like the iron sulfur clusters in the electron transport chain or cytochromes. This 1 electron transfer capability is a unique biochemical superpower, enabling complex redox reactions. Then there is the reduced form, FADH2 or FMNH2. The ring can accept a second electron and a second proton, becoming fully reduced. This form is rich in energy, storing the electrons, and is ready to donate them to another acceptor. It's colorless, signifying the electron-loaded state. This remarkable versatility allows flavoproteins, which are enzymes containing FMN or FAD as prosthetic groups, to participate in an incredibly wide range of redox reactions. They often act as vital intermediaries, facilitating electron flow in complex metabolic cascades, unlike NAD+ or NADH, which are freely diffusible coenzymes. Flavins are typically tightly but non-covalently bound to their respective apoenzymes, forming the stable flavoproteins. This tight binding ensures that the electrons are transferred efficiently and in a controlled manner within the enzyme's active site. Let's look at some of the most prominent members. The first is the citric acid cycle, or the TCA cycle. Succinate dehydrogenase, SDH, also known as complex 2 of the electron transport chain, is a truly unique enzyme. It is the only enzyme of the TCA cycle that is embedded within the inner mitochondrial membrane, directly linking the cycle to oxidative phosphorylation. It contains a covalently bound FAD prosthetic group, which means FAD is permanently attached to the enzyme, making it an intrinsic part of its structure. So the reaction here is succinate plus FAD leads to the formation of fumarate plus FADH2. Here succinate is oxidized to fumarate and the electrons removed from succinate are accepted by FAD, reducing it to FADH2. What is the significance? This is a crucial distinction for your exam. The FADH2 generated here does not enter the electron transport chain via complex 1, unlike NADH. Instead, it donates its electrons directly to the iron sulfur clusters within the complex 2 itself, which then pass them to ubiquinone, which in short is called CoQ or coenzyme Q. Each FADH2 from complex 2 typically yields approximately 1.5 molecules of ATP via oxidative phosphorylation, compared to 2.5 ATP for NADH. Remember, this numerical difference is crucial. It's a favorite for Viva and MCQ questions. Genetic mutations in SDH can lead to rare mitochondrial disorders and even predispose to certain tumors like paragangliomas. Next is, in the beta oxidation of fatty acids, there's an enzyme called Acyl-CoA dehydrogenase. These are the enzymes involved in the fat burning. So these are the fat burners, Acyl-CoA dehydrogenases, and they are involved in beta-oxidation of fatty acids. Now, a family of FAD-dependent enzymes are called Acyl-CoA dehydrogenases. Example: short chain, medium chain, long chain, very long chain, etc. These initiate the beta-oxidation spiral, the pathway that breaks down fatty acids into acetyl-CoA. The reaction is like Acyl-CoA plus FAD gives trans-enoyl-CoA plus FADH2. This is the very first redox step in the cyclical process of breaking down fatty acids. Now the significance of this is the FADH2 generated here is then reoxidized by the electron transferring flavoprotein, which itself contains FAD. This in short is called ETF. ETF passes these electrons to ETF ubiquinone oxidoreductase, which is another flavoprotein, which ultimately transfers them to COQ, coenzyme Q, in the ETC. This is another example of FADH2 entering the ETC at a lower energy level, bypassing complex 1. Now, deficiencies in these acyl-CoA dehydrogenases, particularly medium chain acyl-CoA dehydrogenase deficiency or MCADD, are important inborn errors of metabolism. In India, while newborn screening programs are still evolving, MCAD or MCADD can present as hyperketotic hypoglycemia, lethargy, and encephalopathy, especially during periods of fasting or illness in infants. Early diagnosis, often through tandem mass spectrometry, is crucial to prevent severe neurological damage. Next, we have the pyruvate dehydrogenase complex. This is the gateway to TCA. The pyruvate dehydrogenase complex is a massive multi-enzyme complex that converts pyruvate from glycolysis into acetyl-CoA, linking carbohydrate metabolism to the TCA cycle. Its third component, which is the E3 component, is called dihydrolipoyl dehydrogenase. It contains a tightly bound FAD. So while E3 doesn't directly generate FADH2 for the ETC, it plays a crucial role in regenerating the oxidized lipoamide cofactor, which is essential for the complex's catalytic cycle. It transfers electrons from reduced lipoamide to NAD+, producing NADH. So what is the significance? Although FAD isn't directly feeding electrons into the ETC here, its presence in E3 is absolutely vital for the continuous function of PDHC or PDH complex. If E3 is impaired, pyruvate cannot be converted to acetyl-CoA, leading to a backup of pyruvate and its conversion to lactate, resulting in lactic acidosis. So the PDHC deficiency is a severe and often fatal neurological disorder, particularly in children, presenting with chronic lactic acidosis and development delay. Next is glutathione reductase. This is the antioxidant defender. It is an enzyme which is a crucial FAD-dependent enzyme that plays a central role in antioxidant defense. So the oxidized glutathione with NADPH plus H plus forms reduced glutathione plus NADP plus. Now what is the significance? It regenerates reduced glutathione from its oxidized form, utilizing NADPH as the electron donor. The reduced glutathione or GSH is a powerful antioxidant protecting cells from oxidative damage caused by reactive oxygen species. This is especially important in red blood cells, where constant exposure to oxygen makes them vulnerable to oxidative stress. Now, the activity of erythrocyte glutathione reductase is a widely accepted biochemical marker for assessing riboflavin nutritional status. A high erythrocyte glutathione reductase activity coefficient, or EGRAC, indicates riboflavin deficiency, as the enzyme's activity increases when saturated with exogenous FAD in vitro assays. This is a common test you might see in a biochemistry lab. Next is monoamine oxidase, or in short MAO. It is the neurotransmitter regulator. Monoamine oxidase is a family of FAD-dependent enzymes, MAO-A and MAO-B, primarily found in the outer mitochondrial membrane. It catalyzes the oxidative deamination of monoamines, including neurotransmitters like serotonin, dopamine, and norepinephrine, as well as tyramine. Now, the significance is that MAO is crucial for regulating the levels of these key neurotransmitters in the brain, as well as in the peripheral tissues, thereby influencing mood, cognition, and behavior. MAO inhibitors are a class of drugs used as antidepressants and in the treatment of Parkinson's disease by increasing the availability of these neurotransmitters. Next is the NADPH oxidase, the immune warrior, or in short NOx. It is a multi-subunit enzyme complex where one of its crucial components contains FAD. It catalyzes the formation of superoxide radicals from oxygen using NADPH as the electron donor. This respiratory burst is critical for immune cell function, particularly in phagocytes like macrophages or neutrophils. The superoxide radicals and subsequent reactive oxygen species generated are potent antimicrobial agents essential for killing ingested pathogens. Genetic defects in NADPH oxidase components leads to chronic granulomatous disease, or CGD, which is an immunodeficiency characterized by recurrent severe bacterial and fungal infections due to the inability of phagocytes to mount an effective respiratory burst. Then there is the methylene tetrahydrofolate reductase, or MTHFR. This is the folate link. It is a crucial FAD-dependent enzyme in folate metabolism. It catalyzes the irreversible reduction of 5,10-methylene tetrahydrofolate to 5-methyl tetrahydrofolate, the predominant circulating form of folate. Now, the significance is that 5-methyl tetrahydrofolate is required for the methylation of homocysteine to methionine, which is a crucial step in the 1-carbon metabolism pathway, essential for DNA synthesis, repair, and epigenetic regulation. Polymorphisms in the MTHFR gene are common, especially in the Indian population. Individuals with these polymorphisms may have reduced MTHFR enzyme activity, leading to elevated homocysteine levels, or which is also called hyperhomocystinemia, which is a risk factor for cardiovascular diseases. Other than that, it can lead to neural tube defects and recurrent pregnancy loss. Adequate riboflavin status can help optimize MTHFR activity even in individuals with these genetic variants. Next is the pyridoxine 5-phosphate oxidase, which activates another B vitamin. So, pyridoxine 5-phosphate oxidase, or PNPO, is an FMN-dependent enzyme. It converts pyridoxine 5-phosphate, PNP, and pyridoxamine 5-phosphate, PMP, to pyridoxal 5-phosphate, or PLP, which is the biologically active coenzyme form of vitamin B6. Now, the significance is that without adequate FMN, the activation of vitamin B6 is severely compromised. This highlights the intricate interconnectedness of B vitamin metabolism. In cases of vitamin B6 deficiency, ensuring adequate B2, which is riboflavin status, is important for effective B6 supplementation. In rare cases, PNPO deficiency itself can lead to severe neonatal epileptic encephalopathy. Finally, there is the Cytochrome P450 reductase, or CPR. This is the detoxifier. It is also known as NADPH cytochrome P450 oxidoreductase. It is a unique flavoprotein containing both FAD and FMN. It transfers electrons from NADPH sequentially via its FAD and FMN prosthetic groups to the heme ion of cytochrome P450 enzymes. The significance is This CPR is essential for the function of the vast family of Cytochrome P450 monooxygenases, CYPs, which are critical for the metabolism of a wide range of endogenous compounds like steroids, fatty acids, and exogenous substances like drugs, environmental toxins, xenobiotics. This is a primary detoxification pathway in the liver. Impaired CPR activity due to riboflavin deficiency could potentially affect drug metabolism, leading to altered drug efficacy or increased toxicity. Okay, so now we come to the regulation and control to maintain a balance. The cellular levels of FMN and FAD are tightly regulated, like a well-managed dabba service, ensuring just the right amount of food is delivered. The first concept is product inhibition, the too much of a good thing principle. So the synthesis enzymes, riboflavin kinase and FAD synthetase, which we talked about earlier, are subject to classic feedback inhibition. FMN can inhibit riboflavin kinase, which is its own producing enzyme, and FAD can inhibit FAD synthetase, which is its own producing enzyme. This ensures that when sufficient coenzymes are present, their production is downregulated, preventing wasteful synthesis and maintaining optimal cellular concentration. It's an elegant self-regulating mechanism. Next is substrate availability. So substrate inhibition is the one in which the rate of FMN and FAD synthesis is also directly dependent on the availability of their substrate, dietary riboflavin and ATP. If you don't have the raw materials, you can't build the final product. That is the logic. The next method is hormonal and nutritional control. While less direct, the expression levels and activity of riboflavin kinase and FAD synthetase can be influenced by hormones, example thyroid hormones. Along with the hormones, the overall nutritional status also matters. For instance, in conditions of energy stress, the cell might prioritize ATP for other essential functions. Next is the cellular demand. Tissues with high metabolic activity, example liver, heart, kidney, skeletal muscle, typically have higher concentrations of flavoproteins and thus a greater demand for FMN and FAD. This localized demand can influence local synthesis rates. So we talked about four major mechanisms. First was the product inhibition, the second was the substrate availability, third was hormonal and nutritional control, and finally cellular demand. Next we come to the biological significance. Why riboflavin matters beyond the mitochondria. Riboflavin's impact on human health is profound and multifaceted, touching almost every aspect of physiology. It's not just about energy, it's about life itself. Now first, as we have seen, it's absolutely central for ATP production, driving everything from muscle contraction to nerve impulses, active transport, and biosynthesis. Without adequate FAD and FMN, our ability to generate energy from carbohydrates, fats, and proteins is severely compromised. This leads to profound fatigue and organ dysfunction. Next is the antioxidant defense. By supporting glutathione reductase, riboflavin plays a critical role in protecting cells from oxidative stress and damage. This has far-reaching implications for preventing chronic diseases. Also, it slows aging processes and maintains cellular integrity. Next is the drug and toxin metabolism. Many flavoproteins are involved in the phase 1 and phase 2 detoxification of drugs, xenobiotics, and endogenous toxins in the liver and other tissues. This is crucial for clearing harmful substances from the body. Next is growth and development. This is essential for normal cell growth, tissue repair, and overall development, particularly during periods of rapid growth like childhood and pregnancy. Deficiency can impair these processes. Next is red blood cell integrity. Through its role in glutathione reductase, riboflavin helps maintain the integrity of red blood cells by protecting them from oxidative damage. Deficiency can lead to hemolytic anemia. Next, we talk about the interactions with other B vitamins. This is crucial, often overlooked aspect. That is why we are talking about it. So the first one is vitamin B6 activation. So the enzyme pyridoxine 5-phosphate oxidase, which we talked about, PNPO, which converts pyridoxine to its active form, pyridoxal 5-phosphate, or PLP, is an FMN-dependent enzyme. So a riboflavin deficiency can functionally impair B6 metabolism. Next is folate metabolism. So methylene tetrahydrofolate reductase, which is also called MTHFR, critical for the conversion of 5,10-methylene tetrahydrofolate to 5-methyl tetrahydrofolate, is an FAD dependent enzyme. Riboflavin deficiency can therefore lead to hyperhomocystinemia, which is a risk factor for cardiovascular disease. This is a classic example of how B vitamins are interconnected, like different sections of a grand Indian wedding band. If one instrument is missing, the whole symphony fails. Finally, we come to clinical connections. So ariboflavinosis is when the complete riboflavin machinery fails. This is where biochemistry meets the patient. What happens when our riboflavin is insufficient? Riboflavin deficiency is also known as ariboflavinosis. It is often seen in conjunction with other B vitamin deficiencies, reflecting general malnutrition. In India, where dietary diversity can be limited for certain populations, or where specific cultural dietary practices prevail, this is not uncommon. What are the causes of this deficiency? The first one is inadequate dietary intake. So this is the most common cause, especially in populations with low intake of dairy, meat, or fortified grains. This can be prevalent in regions with economic hardship or specific dietary restrictions. Next is increased demand. So this is seen in pregnancy, lactation, rapid growth, strenuous physical activity. Next is impaired absorption. So this is seen in chronic alcoholism, malabsorption syndromes like Crohn's disease, celiac disease, chronic diarrhea, tropical sprue, and certain medications like phenothiazines and tricyclic antidepressants. Next is increased excretion. Example, diseases like liver disease or hemodialysis. Next is genetic factors. So rare genetic factors in riboflavin transporters of flavokinase can cause isolated riboflavin deficiency, and this in turn leads to severe neurological manifestations. So what are the clinical manifestations, what you'll see in the ward depending on the riboflavin deficiency? So the first one would be oral pharyngeal lesions. These are classic signs. Example, chiliosis, which is fissures and cracks at the corners of the mouth, also called angular stomatitis. These are painful and prone to secondary infection, like the dry cracks you see on a parched riverbed in summer. And the other one is glossitis, inflammation and magenta colored tongue. It is a tongue that looks like it's been painted with gulal, but not in a good way. Next is pharyngitis or sore throat. So these were the oral pharyngeal lesions. Next are ocular manifestations. These include corneal vascularization, which is growth of capillaries into the cornea, leading to blurring of vision, photophobia, and eye fatigue. The other one is cataracts, long-term deficiencies associated with increased risk of cataract. Next, we come to the dermatological lesions. These include seborrheic dermatitis-like lesions. This is greasy, scaly skin lesions, especially around the nose, mouth, and scrotum or labia. It looks like a patch of flaky skin that won't go away. Next, we talk about the hematological. These include normochromic normocytic anemia, often related to impaired iron metabolism, and potentially reduced red blood cell lifespan due to increased oxidative stress. So here we can note that riboflavin is needed for proper iron utilization, so it is related to impaired iron metabolism. Next, we have neurological symptoms, which include severe deficiencies, especially in infants with genetic defects, and it can lead to neuropathy, ataxia, and development delays. Now we come to the diagnosis. So the one way to diagnose is clinical signs, often suspected based on characteristic physical findings. The next one is biochemical tests. These are more appropriate and more specific, if you will. The first one we have is the EGRAC. Okay, we talked about this before. This is the most reliable function test. It measures the activity of the FAD dependent enzyme glutathione reductase in red blood cells before and after adding FAD. It's like checking how much a car's engine improves after you give it the right fuel additive. Next is the urinary riboflavin excretion. This is less reliable as it reflects recent intake more than tissue stores. Finally, you come to the treatment. So oral riboflavin supplements, typically 5 to 30 milligrams per day, typically resolve symptoms within days to weeks. Dietary counseling is also crucial. Now I'd like to talk about a couple of mnemonics and memory tricks. Okay, let's make this stick like laddu to your fingers. Okay, so the first one is riboflavin's color. R for riboflavin, R for reddish yellow. Think of ripe rasgulla. Though it's more yellow, the alliteration helps. Deficiency symptoms. The 3 C's and the M. So the 3 C's are cheiliosis, corneal vascularization, and cataracts. And the M is magenta tongue, which is glossitis, right? So the mnemonic, when B2 is low, your cheer crumbles, your eyes cry, and your tongue turns magenta, like a sad Bollywood movie. Okay, finally, I would like to summarize and give you the take-home message. Here are your high-yield takeaways for your exams and your patients. So the first one is riboflavin is the precursor for active coenzymes like FMN and FAD. Next, FMN and FAD are versatile electron carriers capable of 1 or 2 electron transfers, unlike NAD+ or NADH. Next is, they are central to energy metabolism, especially the electron transport chain, complex 1 and 2, and beta-oxidation. Next is, remember the ATP yield difference. NADH is 2.5 ATP and FADH2 is 1.5 ATP. This is a crucial point. Next, they are essential for antioxidant defense via glutathione reductase and the activation of other B vitamins like B6 and folate. Next is ariboflavinosis presents with characteristic signs like cheiliosis, angular stomatitis, glossitis, corneal vascularization, and seborrheic dermatitis. Lastly, the EGRAC test is your diagnostic gold standard. So I'd like to end my video here. Hope you all understood the intricacies of vitamin B2. Thank you for watching. Hope to see you soon in the next video.